My name is Jocelyn Bell-Burnell. I'm the current president of the RSE, and I'm chairing this evening's sessions. The main piece this evening is the lecture, but we have a couple of rather happy things first. We have a couple of medals to present. I'm going to start by presenting the more junior one, then present the more senior one to the person who also happens to be our speaker, and then introduce the lecture. But before we get to the fun stuff, there's some more serious stuff. Maybe even I shouldn't assume that you all know what the Royal Society of Edinburgh is. Some of you may be here for the first time, and you're particularly welcome if you are. We're sometimes described as Scotland's National Academy. We have about 1,600 fellows. They're the top academics, they're the leaders of the professions, of businesses, civic leaders, practicing artists, musicians, writers, a wonderful range of really outstanding people. And the aim of the Royal Society of Edinburgh is, through all these fellows, to contribute to the social, cultural and economic well-being of Scotland and advance its advancement of learning and useful knowledge, which was part of the Society's original charter. Move to this evening's particular business. And first of all, I have to present what's known as the RSE Patrick Neal Medal, which is the Early Career Prize in Life Sciences. And the 2016 medal is going to Dr. Asier Unsiti Brosetta. The medal's named after Patrick Neal, Dr. Patrick Neal, who lived in the late 1700s, early 1800s, a distinguished Scottish naturalist and fellow of the RSE. He devoted much of his late career to natural history, botany, horticulture. And this year's winner is the PI and group leader of Cancer Research UK Edinburgh Centre, MRC Institute of Genetics and Molecular Medicine, University of Edinburgh. And it's for his outstanding research work in biomedical technologies and innovative therapeutics through multidisciplinary collaboration. So could I invite Dr. Asier Unsiti Brosetta to come forward to receive his medal, please? And our second medal, the Senior Prize Medal in Physical Sciences. Physical Sciences? Not sure about that, but anyway. Senior Prize is the RSE Sir James Black Prize Medal, and it's being presented this evening to Professor Thomas Simpson. It's named after Sir James Black, one of many distinguished James Blacks. He died in 2010. He's a Scottish doctor and pharmacologist. He was a Fellow of the Royal Society of London, an Honorary Fellow of the Royal Society of Edinburgh. He won the Nobel Prize for Medicine in 1988, the Order of Merit in 2000, a knighthood in 1981, and here comes it, and the RSE Royal Medal in 2001. And we're very pleased to have Rona Black, his widow, with us today. So the 2016 winner of the James Black Medal is, as I've said, Professor Thomas Simpson, Alfred Pass Professor of Chemistry, School of Chemistry, University of Bristol, for his outstanding contribution to the biosynthesis of natural products as a pioneer in the interdisciplinary field of chemical biology. So would Professor Thomas Simpson come forward to receive his medal? And then we move on to Professor Simpson's lecture. The title is New Drugs from Old Pathways, Teaching Nature New Tricks. Professor Simpson started life in Alloa, did a BSc in Edinburgh, PhD in Bristol. He's worked on the chemistry and biosynthesis of biologically important microbial natural products. There will be some opportunity for question and answer at the end of the talk, but I'll leave that till then. Would you now welcome, please, Professor Thomas Simpson to speak about new drugs from old pathways, teaching nature new tricks.
Hope we're going to have some pictures here. Yes. Okay, well, it's obviously a great honour and pleasure to be here tonight. I'd like to thank the Royal Society of Edinburgh for selecting me for this very special award, and also the President for her kind introduction. I should also congratulate my colleague here on the Junior Award. It's always nice to see the chemists putting one over on the biologists. Usually the other way around. So that's the title of my talk, New Drugs from Old Pathways, Teaching Nature New Tricks. And what I want to do this evening is try to describe how we can harness through molecular genetics the pathways by which nature makes biologically active compounds, particularly in microorganisms, fungi and bacteria, with two general aims. One, to increase the molecular diversity which is available to us, and in a second part, to show how we can perhaps home in and refine the properties of an existing clinically used antibiotic. But first I must thank two people. First of course, Sir James Black, who was a very wonderful and very special scientist, and obviously who's lent his name to this medal. But I also want to take the opportunity, there he is there, but I also want to take the opportunity to say thanks to someone else, and that's my PhD supervisor and lifelong mentor, Jake Macmillan. Partly because you'll see there they have quite a number of things in common, besides an interesting taste in spectacles. They were both born in Lanarkshire in 1924. They spent formative parts of their careers in the University of Glasgow and also with ICI. And they were both elected Fellows of the Royal Society within a couple of years of each other. I think they were both very much ahead of their time in applying logic and design to the discovery of new biologically active entities. And obviously, Sir James is responsible for the discovery and development of two of the biggest selling ever drugs, which is some achievement. Jake was also interested in biologically active compounds. He isolated griseofulvin, a still clinically used antifungal agent, when he was a very young researcher at ICI, fresh out of his PhD, and there's interesting stories related to that. But the focus of his career, lifelong career, was the gibberellin plant hormones. And I should say that Jake Macmillan was doing what we now know as synthetic biology some 40 years before it became the kind of trendy area now beloved of research councils. I have to say that because we need money from them. He was carrying out, throughout his career, he carried out interdisciplinary research, working with biologists, microbiologists, geneticists. And that's the lab I entered as a young PhD student more years ago than I care to remember. But despite his lifelong involvement in interdisciplinary research, Jake always regarded himself first and foremost as an organic chemist, as do I. And of course, as chemists, that means we're fascinated by, I hate to tell the non-chemists in the audience, molecular structure, how it's elucidated, how it can be modified, synthesized, and in our case, how it can be biosynthesized, the pathways by which living organisms produce these very interesting compounds. And we'll have more of that in a moment. The background to this talk is, of course, the antibiotic crisis. And as the slide shows there, there's been a steady decline, you might say a catastrophic decline, in the number of new antibiotics coming onto the market for many decades. And of course, this has coincided with the very rapid increase of resistance to a number of very important pathological bacteria. For a number of reasons, some of which are listed here, Big Pharma has reduced its efforts or its investment in discovering new antibiotics. Now, this isn't meant to be a critique of the pharmaceutical industry. The pharmaceutical industry is an industry. It's not a charity. And like every other industry, it's subject to the dictates of the stock exchange. No profits, no industry. But I also think we as the general public have a role in this as well. While we urge and indeed demand increasing really vast amounts of money on very expensive anti-cancer drugs, which at best might prolong life or quality of life for a few weeks or a few months at best, we expect antibiotics to be cheap as chips. And these are compounds which actually save life. So there's a real dichotomy there that I think is something that we should all pay attention to. So there is a real problem here. You can tell it's a problem if even Jeremy Hunt realises it's a problem. And of course, other authorities such as the World Health Organization and other health authorities have recognised this. And of course, they're aware that this can't be left to the market alone. And new strategies must be developed for developing new antibiotics, which are urgently needed. Now, about a third of all drugs in clinical use are actually natural products themselves, or are derived by or inspired by natural products. And this rises to almost 2/3 in the case of anti-infective and anti-tumour agents. So this is an important source of biologically active compounds. Now, as we'll see, one of the problems with natural products is their inherent structural complexity means it's not easy to adopt the traditional synthetic or medicinal chemistry approach to modifying and refining their structures and their properties. But in recent years, with the isolation of biosynthetic gene clusters, particularly through whole genome sequencing, which is now very rapidly available, and the development of molecular genetic techniques, this provides us with a real opportunity for modifying these existing pathways to produce new compounds and compounds with optimized properties, both in terms of activity, but also in production. But why are natural products effective as drugs? They have generally high molecular weights, quite complex structures, and they tend to be highly polar. This means that they contravene what is known as Lipinski's rules. Now, Lipinski's rules were formulated by Chris Lipinski, who was a pharmaceutical chemist, who essentially analysed the structures of known synthetic drugs. And he was able to observe that most drugs are relatively small and lipophilic, which quite the contrast to natural products. And he formulated his rules of 5 for druggability. The molecular weight shouldn't be more than 500. You should have no more than 5 hydrogen bond donors or acceptors. And this important aspect here, oops, go back. The partition coefficient should not be greater than 5. And that's really just a measure of its solubility in lipophilicity. But fortunately, there is a 5th rule. And the 5th rule states that none of the above apply to natural products. And one has to question why. Natural products are said to be privileged structures. And of course, as they are made by enzymes, by proteins, one might expect them to be able to react or interact readily with proteins, enzyme active sites, receptors, and the like. And indeed, that is the case. And so I've been warbling on about natural products, and most of you probably don't know what I mean by a natural product. So what are they? Natural products are compounds produced by living organisms, microorganisms, higher plants, marine organisms, and the like. And they're produced by a process known as secondary metabolism. Now, primary metabolism is essential biochemistry, which is common to almost all living organisms, from a bacterium to an elephant. They have common biochemical pathways. But many organisms have what are called secondary metabolism, where using basically the same building blocks and many of the same types of enzymes, they can produce a wide range of what we now call secondary metabolites of natural products. The interest has been that these compounds, although they have no obvious role in the economy of the organism which produces them, they have properties which man, for generations, indeed for millennia, have found of interest. Antibiotics, drugs, flavours, poisons, narcotics, and all the rest of that. And that's best illustrated by giving you a few simple examples. This simple unsaturated alcohol is called geraniol, which is one of the major constituents of rose oil. So when you smell a rose, you're smelling some of this compound. Lycopene is a naturally occurring pigment responsible for the bright red colour of tomatoes and red peppers, etc. So these compounds bring nice smells, nice colours into our life. A compound which has probably been of interest to Sir James is digitalin, the heart-stimulating compound produced, of course, by Digitalis purpurea, the purple foxglove. Another compound much used and, of course, abused by man is morphine, produced by this very attractive plant here, the opium poppy, Pabava somniferum. And many of you, on your way to and from Tynecastle and Murrayfield, might have passed a factory which goes under the name of Macfarlane Smith and probably wondered what it is. Well, Macfarlane Smith is, in fact, the world's major producer of opium and opiate-derived drugs. I actually got the Macfarlane Smith Prize for chemistry when I was a student, and it was 20 years afterwards I discovered what it was all about. I should say the world's largest legal producer. I'm sure there's a few rivals. And then, of course, there's antibiotics. This is penicillin G, produced by Penicillium chrysogenum, which really kicked off the whole antibiotic story. And this is Penicillium chrysogenum growing in what are formerly known as penicillin flasks, but more colloquially known as bedpans. And these came from the original wartime production plant for penicillin in Liverpool, in Speke in Liverpool, which at the time was the largest in the world. I spent the early days of my PhD using these flasks and growing up organisms. And I can well remember as a young PhD student on a hot afternoon in Bristol, making up the medium for these and then sterilising them over a hot, sweaty autoclave. It wasn't really my idea of what research chemistry was about. It was a way of passing the time. And, of course, the Nobel Prize for Medicine in 2015 was awarded to Satoshi Omura from the Kitasato Institute in Tokyo and the wonderfully named UU2 from the Chinese Academy of Science, Traditional Medicine, for their work on the discovery of and development of antiparasitic compounds, ivermectin isolated from this compound, Streptomyces, this organism, Streptomyces avermitilis, in the case of Omura, and UU2 was responsible for really developing and optimizing the maximum yields of the important antimalarial compound artemisinin. So hopefully this shows that natural products are indeed an important source of valuable compounds. Now, what attracted me to natural product chemistry in the first place was that it gets you involved in a whole range of activities. First of all, you have to isolate and purify the compounds, and that isn't a trivial exercise. And once you have the compound pure, you can elucidate its structure. Still one of the things I get a major kick out of. Once you have the structure, you can then begin to speculate on how this might be biosynthesized, and then carry out biosynthetic studies of different levels of sophistication. In the early days, it was incorporation of simple precursors to identify the basic building blocks. incorporation of more advanced intermediates, which again was a synthetic challenge. And of course, one needs to unravel the enzymology and the genetics, the genes, of course, which encode the enzymes. Natural products are major targets for synthetic chemists. I do delight in telling my synthetic colleagues, if it wasn't for the poor bloody natural product chemist isolating interesting structures, they would have nothing interesting to synthesize. They don't always agree with that. And of course, what I'll be talking about is can we manipulate these pathways? Now, to look at this in a slightly different context, here's another product. Some of you may find that desirable, others less desirable. But if you were to ask yourself, how is this compound produced? What questions would you ask yourself? So we can compare biosynthesis with manufacturing process. And so you want to ask, what are the basic components, the basic building blocks? What order are they assembled in? Does it actually matter? What equipment, how are these components assembled in the right order? And what machinery do we need for this? And to ask more sophisticated processes, what controls the overall process? Delivery of the starting materials at the right time, how much manpower do you need, and obviously what do you do with the product once you have it. An important question in these incipient Brexit days, where do we site the factory? Probably not Sunderland. And also, can we make hybrid compounds? For example, I put an example there. I was going to suggest putting a Honda engine and a McLaren chassis on Pirelli wheels, but a certain Mr. Alonso thought that might not be a very good idea. So, the compounds that I'm interested in are produced by a pathway called the polyketide pathway. And all I want to illustrate here is the sheer range and variety of structures, all the way from simple aromatic compounds through to really quite complex polyfunctional polycyclic molecules. And the other thing, of course, is many of these compounds have important biological activity associated with them. And even these compounds alone have annual sales of hundreds of billions of dollars. So these are commercially and economically very important compounds as well as what they do in terms of healthcare. Down here we have lovastatin, which is the progenitor of the statins. Now, I've been out of the country for 2 weeks, but last time I was here, the official line was that statins are good for you. That might have changed in the last 2 weeks. I believe they're good for you, but I'm perhaps biased. So how are these compounds produced? And I'm going to tell you two main stories. One, how we can manipulate a fungal polyketide, non-ribosomal peptide synthase, I'll explain what that is in a moment, to really create enhanced molecular diversity. Now, one can't really predict biological activity, but the more compounds and variations and structures you produce, the more chance you have of finding compounds which will do something useful. So the first is really going to be an exercise in creating molecular diversity. And the second is engineering the pathway to a currently clinically important bacterial antibiotic to see whether we can overcome the limitations that are inherent in its use. So let's start with fungal polyketides. Now, for those of you in the know, the polyketide pathway is very closely related to a corresponding pathway in primary metabolism, that of fatty acid biosynthesis. And the basic pathway starts from acetate, a simple 2-carbon unit. And what happens is these 2 carbon units are linked to the synthase. I'll say a bit more about that. So acetate plus malonate, and these solid lines, which you'll see in a number of the slides, indicate pairs of carbons derived from an original acetate unit. So these two condense together under the mediation of an enzyme called a ketosynthase to make this beta-ketothiol-ester. It produces a ketone, therefore it's called a ketosynthase. Biochemists are very simple-minded people. They tell it how it is. This may or may not be methylated at this stage, and from using an enzyme known as the C-methyl transferase, which just simply adds this methyl group. You can then reduce the ketone to an alcohol using a ketoreductase. You can then remove or eliminate the water with an enzyme called a dehydratase to introduce this double bond, and then you can further reduce that double bond, produce a saturated compound. And that's one assembly cycle. And what polyketide synthases do is they use this basic cycle in an iterative manner. But the real trick, and as I'll elaborate on, is in each cycle, it can use different combinations of these activities to make polyketide chains of a huge variety of functionality along the chain. Now, fungal polyketide synthases come in a variety of forms depending on the complexity of the molecule they produce. I'll be talking about this one at the bottom here, which is called the highly reducing type 2 polyketide synthase. And what this is a single large protein encoded by a single gene, but that protein is folded into a number of distinct catalytic domains. And you can think of these as beads on a string or as Lego blocks. I'll probably use the Lego block analogy. So we have these Lego blocks, each of which catalyzes a different chemical event. These are very large proteins, molecular weights of up to 3 million. As yet, we know very little for sure about the actual detailed structure of these polyketide synthases, other than the fact that they do exist in these discretely folded catalytic domains. But a few years ago, what we do know from sequence comparison is that these fungal polyketide synthases are probably very similar in structure, as well as their basic biological activities, to mammalian fatty acid synthase, the structure of which was determined by Ban and co-workers in Zurich about 10 years ago. And what we have along the bottom here is what we call essentially a polymerase. So this is the ketosynthase, the acyltransferase, and acyl caroprotein, which simply link the acetate units together. And then at the top, we have the domains which are responsible for modifying this chain, the ketoreductase, the methyltransferase, and so on. And just to prove that chemists and scientists are not totally humorous, this structure has been dubbed, for reasons which may or may not be obvious, the gingerbread man structure. And use your imagination, and you might be able to see some analogies there. Now, the system I'm going to talk about initially is this compound here called Pennilyn, which is produced by a fungus called Beauveria bassiana. And some of you might recognize this name here. This was early work by David Hagen, along with what I believe was his first PhD student, Russell Cox, again I shall mention. And using classical isotope labeling experiments, we were able to show that this part of the molecule was derived from the common amino acid tyrosine, but this part was polyketide. And what we have here is what I'd call a pentaketide. If you count the bars, there's 1, 2, 3, 4, 5, indicating this is derived from 5 acetate units with the introduction of 2 C-methyl groups, and therefore this would refer to as a dimethylated pentaketide. And I'll be using this nomenclature quite a bit in the next few minutes. You can also see that this does nasty things to insects. And there's even been a proposal that this is used as the spores of this organism are used as a treatment for bed bugs. You just make a suspension of them, spray it on your sheets, and no longer got itchy bites in the morning. It was in the Daily Mail, so it must be true. Now, we were able to isolate the gene, or the genes responsible for the production of Pennilyn. There's actually 3 genes. One of them is this encodes this very large protein here, where we can see the highly reducing polyketide synthase, but it's linked to the domains characteristic of a non-ribosomal peptide synthase. Now, non-ribosomal peptide synthases, or NRPSs, are responsible for activating amino acids and linking them together via amide bonds. And really, these are amazing, fascinating molecular machines. And what I want to do, just fairly briefly, is go through what this actually does, how this compound is actually made. So things will move fairly fast. So the first thing that happens is acetate and malonate get added to the synthase. They're then condensed by the ketosynthase. You then get a C-methylation. You then get a ketoreduction, a dehydration, and an enol reduction. So here, in this first assembly cycle, we're seeing all the activities of the synthase utilized. We then go on to a second cycle, where we have much the same happening, except we don't have the initial or the final enol reduction, and so we retain this carbon-carbon double bond. There's then a third cycle where there's no methylation occurs, but dehydration or ketoreduction and dehydration does occur, and then a fourth cycle where we quite simply get a chain extension, the ketosynthase. At this point, the polyketide synthase has done its job, and the NRPS domains take over, and they have the job of activating tyrosine and adding it to this so-called thiolation domain. The condensation domain then forms an amide linkage with the polyketide part, and then right at the end there's this something called a DKC, which in the trade is known as a Dieckmann cyclase, and it catalyzes formation of a carbon-carbon bond between this activated position and this activated position to form a 5-membered ring. And in the process, this compound known as pre-Pennilyn A is released from the enzyme. So this is the first enzyme-free intermediate in this process. And for those of you who haven't been counting, there's actually 16 enzyme-bound intermediates in the process of making this. And of course, we can't really capture them or interfere with them in any way. There's then 2 oxidative processes. 10A, which is a P450 enzyme, oxidatively expands this 5-membered ring to form a 6-membered ring. And finally, 10B causes hydroxylation of this position to find the final compound. And if we knock out 10B or 10A, we can isolate pre-Pennilyn B and pre-Pennilyn A as new natural products. So we can ask a number of questions. I've alluded to what controls the process of polyketide chain assembly. And I would posit that this is actually one of the major problems in certainly in structural biology and biochemical catalysis. What controls polyketide chain assembly? The kind of questions we ask: what controls the chain length, the actual number of assembly cycles which occur on this iterative protein? And remember, it's a single protein which is used in each iterative cycle. What controls the degree of reduction in each cycle? So to what extent does this ketone group get reduced and eliminated from the system? And the third question we can ask is what controls the degree of methylation in each cycle? Do you get a methylation or not? Do you get 2 successive methylations or whatever? And of course, more interesting in these questions is, can we control them? Because if we can control them, we can make, as we'll see, new molecules. Now, what we chose to do was to adopt a purely empirical approach and isolate the genes associated with the biosynthesis of a number of close related compounds, just look at the sequence, do some bioinformatics, and see if there's any information present in that sequence which would give us a clue to how this control is exerted. This is Pennilyn, as you can see, interestingly produced by Beauveria bassiana, and as I've indicated, this is a dimethylated pentaketide. Another compound in the literature this time is a compound called bassianin, interestingly produced by Beauveria tenalis. I'm not quite sure what happened to the taxonomy between these 2 organisms. But this is a longer chain. It's now a dimethylated hexaketide. So penta is short. I'll use that terminology. Hexa is long. I'll come to that in a moment. This is militarinone, another related compound, which is actually produced by a heptaketide. So 7 acetate units here and 3 C-methylations. So you can see a lot of variations on a basic theme. So we set about trying to isolate bassianin from Beauveria tenalis. This proved to be a fruitless exercise. We looked at about 40 different strains of Beauveria tenalis and related organisms, but none of them produced bassianin. So to all intents and purposes, this is an extinct molecule that no longer exists in nature, at least to the best of our endeavours. But what we did isolate from one strain of Beauveria bassiana was this compound here. And if you compare this with bassianin, it's the same chain length, but only has 1 methyl. So this is produced by a monomethylated hexaketide. And so we set about comparing these 2 systems, which differ in chain length and which differ in degree of methylation. And this again just summarizes it. This is Pennilyn. So remember, blue is Pennilyn, blue is short dimethylated, and pink is desmethylbasianin, and this is long monomethylated. And so we've been able to isolate the genes for both of these systems and express them. And I wasn't going to talk about how we do these experiments, and I'm not really going to, but how do we actually study these things? Again, just remember, it's a single protein with a number of discrete catalytic domains encoded by a single gene. And the DNA encoding discrete domains can be systematically swapped between these different systems. The gene can either be cloned from the producing organism, or these days it's sometimes easier just to synthesize it de novo, and we express it in a heterologous host. A heterologous host means a different factory, where we know that these hybrid systems will be expressed both efficiently and functionally. And what we use is an organism called Aspergillus oryxi. So this is our new factory. And gratifyingly, these hybrid PKSs often produce titers or produce compounds in higher yield than the native producing organisms. So what we're going to do is play with Lego blocks. This is the Pennilyn PKS NRPS, and it produces this short dimethylated polyketide. So remember, blue is short di, and pink is long mono. So what we're going to do is just sequentially replace these blue domains with the corresponding pink domains. And we replace up to three of them, and there's still no change in phenotype. We still see the same structure. But when we replace the methyltransferase, so we've put the desmethylbasianin methyltransferase in this Pennilyn background, something else happens. And what we see is a short monomethylated compound. And this is a new compound. So this is a new compound that we've produced. We then go on successively replacing them. Once we replace the ketoreductase and the ketoreductase and ACP, we now have the pure basianin phenotype, a single, long, monomethylated polyketide. And this implied, rather surprisingly to us, that chain length is controlled by the ketoreductase. And this is a totally unexpected result, but it's only when we replace the ketoreductase that we get a change in chain length. So we set about doing some more selective, targeted replacements. And when we just replace the going backwards. When we just replace the C-methyltransferase, not surprisingly, we get monomethylation, but we retain the short chain. If we go on and add the ketoreductase, again, we go back to the long monomethylated chain. And the really interesting experiment was when we simply put the basianin ketoreductase in the Pennilyn background. Now, what we get is a mixture of 4 compounds, but if we analyze these, what we're getting is predominantly a long chain. We add these 2 together, and we're getting predominantly dimethylation if we count the blue and the beige together. So we really are seeing new compounds here. And I'll show you this same experiment. And again, this really confirmed that the chain length is actually controlled by the ketoreductase. And I think at the outset, if you put your money on the ketoreductase being responsible for controlling chain length, you'd have lost your shirt. So this is the same experiment. We can see it in a slightly different way. This is the HPLC trace of the extract from this hybrid PKS. And what we can see is pre-desmethylbasianin. We've not got the oxidative enzymes present, and so we get this 5-membered ring, a monomethylated hexaketide. But what we also see is a slightly major product is pre-basianin, and this is a dimethylated hexaketide. So what happens when you add the other genes. And so what we've done is we've added the two P450s, which are responsible for ring expansion and hydroxylation. We get this upper trace. And what we see here again is desmethylbacianin, but fascinatingly, what we also see is bacianin. And so remember I said this compound, as far as the world was concerned, was extinct. And so, if you like, we've raised this compound from the dead, and as a result, we've called this a Lazarus metabolite. This was a great pleasure to one of my collaborators, Colin Lazarus, who didn't object to this terminology. Now, just to really finish off this section, we've shown that indeed the ketoreductase is responsible. Militarinone produces an even longer chain, a heptaketide, and so when we put the militarinone ketoreductase in a desmethylbacianin background, sure enough, what we see is monomethylation, but a longer heptaketide chain. And this is just a busy slide, but it's really just to show that, that we can make a huge variety of structures just by simply varying these basic components. So we can make penta, hexa, or heptaketides, we can make tetramic acids, 5-membered rings, 6-membered rings, hydroxylated 6-membered rings. And there's about 30 structures there, and we've been able to produce about 20 of these by these types of genetic manipulations. So here we really are generating great structural diversity by manipulating these basic systems. And of course, the majority of these are novel new compounds which have not been seen before. So we can create molecular diversity. So there's the basic questions. We said, can we control them? And I guess the answer is, yes, we can. But why we can is a completely different reason, different question. Give me about £10 million in 5 years, we might be able to come up with a definitive answer to that. It's fundamentally a problem in substrate protein recognition. And we are trying to address that in a variety of ways. So the second story, engineering the pathway to an important bacterial antibiotic to improve efficacy. No idea how I'm doing for time here. So let's look. Oh, what's happened here?
I think you may have blanked it, excuse me.
Oh, I see, all right. Glad someone here understands the technology. So what I'm going to talk about in the rest of this talk is the antibiotic called mupirocin, which is isolated from Pseudomonas or fluorescens. Structurally, this is a fascinating molecule. I won't bore you with too many details, but we have this entity here called monic acid, which is polyketide in origin. We have a fatty acid derivative here. And these are linked together to form a molecule called pseudomonic acid A. The generic term is mupirocin. It's the active component of an antibacterial skin cream called Bactroban. This is the thing for teenage acne, which I suffered from dreadfully, but this came along about 20 years too late to give any benefit for me. It's also used widely as a preoperative nasal spray to combat MRSA infections, obviously in operations. It works by inhibiting an enzyme called isoleucyl tRNA synthase. And here we have the crystal structure of the isoleucyl tRNA synthase with pseudomonic acid bound in it. Although it's a long linear molecule, you can see that it kind of curls up in the active site. And what it does, it prevents the enzyme from loading isoleucine onto the corresponding tRNA and therefore disrupting normal ribosomal protein synthesis. So we do understand how this works. Now, the limitation with mupirocin is that the molecule is inherently unstable, and that's why its use is limited to topical or infections or nasal sprays. And the reason that it's chemically unstable is due to this moiety here. For those of you who don't know, this is called an epoxide. It's a very strained 3-membered oxygen-containing ring, and it's very subject to being ring-opened by nucleophiles. And here we have a nucleophile, And so this molecule can essentially bite itself either at this end or this end to produce these two compounds, both of which are inactive. And so out with a narrow pH range, typically in serum, the compound is very soon rendered inactive. Now, in fact, mupirocin is a mixture of three compounds. I've shown you pseudomonic acid A, which has this epoxide group, and it constitutes about 95% of the preparation. There's a few percent of a compound called pseudomonic acid B, which differs from pseudomonic acid A in having this hydrogen replaced by an OH group, very apparently trivial modification. And then there's a trace, literally trace amounts, of this compound called pseudomonic acid C, which lacks the epoxide and has simply a double bond. Now, I'd wager there's a number of people in this audience who do know a little bit about biosynthesis. I wager if I was to ask them the biosynthetic relationship between these three compounds, they'd say that pseudomonic acid C is a precursor to pseudomonic acid A, the double bond being epoxidized, a very facile process in biosynthetic systems, and then pseudomonic acid A would be further hydroxylated to form pseudomonic acid B. So the pathway should be C to A to B. You can tell from me I'm talking that that's not going to be the case. And we also tested these for biological activity. And interestingly, there's nothing in the literature about the relative activities of these. Turns out that pseudomonic acid A, and there's a simple plate assay, we can see a large inhibition zone for pseudomonic acid A, so it's clearly active, despite the fact it's relatively unstable. Pseudomonic acid C is at least as active, and of course, chemically, this lacking the epoxide is a much more stable molecule. Surprisingly, pseudomonic acid B, where we just put this extra hydroxyl group, is essentially inactive. And so this is one of the issues, that a very simple change in structure renders the compound inactive. And so we've posited that pseudomonic acid lacking this reactive epoxide should be more stable and have much wider applications as an antibiotic. So that begs the question, can we knock out the gene responsible for introducing this epoxide so that pseudomonic acid C becomes a major metabolite? So if you simply block the pathway after pseudomonic acid C, theory tells us that pseudomonic acid C should accumulate and become the major metabolite. This is the mupirocin biosynthetic gene cluster. We sequenced this, 13, 14 years ago now, and I should say this is a collaboration with Chris Thomas in Birmingham. And I won't go into the detail of this, but one thing you can see, there's a hell of a lot of genes there. And there were far more genes present in the gene cluster than we thought would be necessary to make this compound. Now that was then, as I say, you Ken knew that it's a very different story. This is an extremely complicated biosynthetic pathway. Now, again, picture, this is HPLC of an extract of the wild type of Pseudomonas fluorescens. I should say the conditions of growth here have been optimized to give us a very clean extract so that we can see all the minor metabolites. Now, this is pseudomonic acid A, and as remember, that's about 95%, so that peak should be way up through the ceiling. We can see pseudomonic acid B, which is about 3 or 4%, and down here, you can see pseudomonic acid C. So you can see that it's a tiny component of the mixture. We've elucidated the structure of all the other compounds, and they fall into two classes. There are those compounds, like pseudomonic acid A, which contain an epoxide or functionality derived from that epoxide. That's all you need to know. And this actually constitutes about 98% of the extract. And then there's pseudomonic acid C, and as you can see, some other compounds containing that double bond, non-epoxidized compounds, and they are less than 2% of the total extract. So what happens if we identify and knock out the gene responsible for the epoxidation? Well, again, here's the wild-type extract. This is what happens when we knock out the gene. We identified this gene, MMPE-OR, that's for oxidoreductase, which is responsible for the epoxidation. This was not an easy task, but we eventually discovered it. And what you can see is all that remains are the compounds derived from the non-epoxidized system, and all the epoxidized ones have disappeared. However, the yield has not been increased. So we've done absolutely nothing to increase the yield of pseudomonic acid C. Another surprise we actually carried out a systematic study of mutating all these biosynthetic genes, growing up the mutant organisms to see what they produced. And we had a major surprise because there's these 4 apparently unrelated genes, which MUP-O, MUP-U, MUP-V, and MACPE, which encode these different activities. And what we found was knocking out any of these caused a complete switch from pseudomonic acid A production to pseudomonic acid B production. Now, any of you know anything about genetics, this is a rather curious observation, because normally if you knock out a gene, you should knock out function. What we've got here is we're knocking out genes and we're getting an apparent gain of function. We're making a more complex molecule. So there's a conundrum here. So how do we explain this? Well, this was our originally proposed, as I said, biosynthesis, C being epoxidized to A, being further hydroxylated to form B. But what these experiments are in fact telling us is that pseudomonic acid C is actually produced by a shunt. So somewhere along the pathway, this pathway to pseudomonic acid C branches off, and so it's not involved in these 2 at all. So C is not a precursor to A or B, and what this also tells us is that B must be an intermediate on the pathway to A. And so somehow we have to lose that hydroxyl group, lose function. And again, I don't want to dwell on this, but we know that this system here, this methyl group, has to be converted to this epoxide, and we form the 6-membered ring by ring opening of this epoxide, giving us this hydroxyl group here as part of the ring-forming process, and we know that this ring is essential for activity. So we've actually, just go back a bit, so we've actually studied in detail the conversion of B to A, which is quite simply losing that oxygen atom and converting that hydroxyl group to a hydrogen. You think that might be a relatively simple process. This is just the cover from Chemical Journal Angewandte Chemie, where we drew this cartoon to indicate. So this is pseudomonic acid B with its hydroxyl here. This is pseudomonic acid A, if you like, are 2 beaks. And what we said, we have a very long, winding road in getting from B to A. In fact, when you look at it, simply loss of hydroxyl, but requires 7 genes, 5 intermediates, 2 of which are enzyme bound, and only the last, pseudomonic acid A, has got significant biological activity. Now, this is probably telling us something about the physiology of the organism. It's a bacterium producing an antibacterial compound, so it doesn't want to produce it until right at the end of the story. And there are other examples of this. So pseudomonic acid A is the active component, and it produces it right at the end of what is a very, very complex pathway. Pseudomonic acid C, as I said, is active, but it's produced in very tiny amounts, so it's not a problem. So this begs the question, can we shut off this pathway and divert everything to pseudomonic acid C? So how can we do this? So I've already indicated mutation of the epoxidase leads to very low titers. And this leads me to a related compound called thiamarinol. Thiamarinol, as you can see, is a structurally related compound. It's produced by a different bacterium, in fact, a marine bacterium, Pseudomonas, and it differs from pseudomonic acid in a number of ways. It has this extra hydroxyl group here. It has an 8-carbon chain here instead of a 9-carbon chain, and we know that chain length here has an influence on activity, and it has this bicyclic, heterocyclic system attached as an amide, whereas pseudomonic acid is a free carboxylic acid. We've synthesized we've sequenced the biosynthetic gene cluster for thiamarinol, and although there's some swapping around, essentially all the mupirocin genes are present in thiamarinol with an extra set of genes which are responsible for producing this moiety here. The other thing is, significant difference, is pseudomonic acid A has the epoxide, thiamarinol A has the double bond. And what we know is that this system here produces thiamarinol A efficiently and in high titer. So that tells us that some of the downstream genes and the proteins that they encode can accept, or preferentially accept, in fact, this double bond, whereas in mupirocin, the same intermediates preferentially accept the epoxide. And so that begs the question, can we make a hybrid polyketide synthase combining the mupirocin and thiamanol genes to make pseudomonic acid C? Now, for a variety of reasons, I can't go into the details of what we've done here, but basically we've taken the epoxidase mutant and replaced various of the downstream mupirocin genes with equivalent thiamanol genes. This is HPLC trace of the crude extract, and what you can see is the major compound is now pseudomonic acid C. This is very readily purified in a couple of Cephalodex chromatography steps to give us essentially pure pseudomonic acid C. For the cognoscenti, this is just comparing the authentic NMR of the authentic compound with the new compound, the compound we've isolated. And the big thing is we have titers of more than 50 milligrams per litre. So these are now comparable with the major mupirocin metabolites. So we've in some ways achieved our major goal here. Hopefully this compound will have much wider clinical application because of its stability than pseudomonic acid A. Of course, because mupirocin is a mixture which contains pseudomonic acid C, has been through all the regulatory process, it should be fairly easy to get this compound tested and its potential. So I think my time is running to an end. So just some conclusions. So one is, yes, we can manipulate biosynthetic pathways both to create great structural molecular diversity, and we can manipulate pathway to select for a particular compound. But there's a caveat. We need to remember a couple of things. First of all, we simply cannot predict antibiotic or indeed any other biological activity. We cannot design an antibiotic de novo. You still have to screen and assay. There's no way of predicting what compound might or might not be active. And because, as I've shown here, even very small, apparently trivial changes in structure can have drastic effects in either gaining or losing bioactivity. So when you hear people talking about designing drugs de novo, ab initio, this has to be taken with a pinch of salt. Anyway, this has been a lot of fun along the way, and I just want to briefly acknowledge some of the people involved. You've seen some of the names earlier. This has been the pseudomonic acid and mupirocin story. There's been a collaboration between Bristol and the University of Birmingham. The fungal work was a long ongoing collaboration with my erstwhile colleague Russell Cox, who 3 years ago decided it was a good idea to go to Germany. He's now frantically learning German, so he can apply for German citizenship. These are some of the fundings, and up here I was being slightly rude perhaps about synthetic biology, but all this work is done on the auspices of the BBSRC-funded Bristol Centre for Synthetic Biology. So I have to admit that I'm now no longer an organic chemist, I'm just yet another synthetic biologist. And I'd like to finish with this slide. This is a major landmark in Bristol, perhaps the only landmark in Bristol, the Clifton Suspension Bridge. This, of course, was a triumph of Victorian engineering designed by Isambard, the wonderfully named Isambard Kingdom Brunel. Unfortunately, didn't live to see its completion. I actually live about 100 yards from this, and so I quite frequently walk across this. And I started off, I don't know if you noticed my first slide, a background of a big mountain, Leotac, and I was going to make the analogy between research and mountain climbing, that research can and probably should be hard work. And you can also make analogies about crossing bridges, etc. And I think in all research, there's a mountain climbing aspect, there's a crossing bridge aspect, and I'm a strong believer that in research, the journey in some ways is far more important and interesting than the destination. So that's something to hang that point on. So again, I must thank the Royal Society of Edinburgh and Sir James Black for this wonderful recognition. and I'd like to thank you all for coming here tonight. I didn't know I had so many friends. Thank you very much. Yes. I'm not sure I can answer them.
No, that's fine. If you need any water in it,
Yes.
water on the table. So our speakers kindly agreed to take questions. If you have questions, could you raise a hand and I'll direct a microphone to you. So, are there questions following a wonderfully clear talk? As a non-chemist, I was very grateful for the hours of work that must have gone into your diagrams, an enormous help for those of us who aren't specialists in the area, and hugely added to the clarity of the talk. So thank you for that. Questions near the back? Thank you.
Tom, from a physical chemist, it's an obvious question, but what about the techniques you use to determine all those structures? I mean, incredible number of structures.
Oh, well, Rob, essentially it's NMR, which as a technique has come on in leaps and bounds over the years. So essentially NMR, sometimes X-ray crystal structure. One of the problems with these molecules are linear molecules with lots of stereocentres, and that makes it quite difficult to establish both the relative and absolute configuration. But essentially, it's just very sophisticated NMR techniques. I did say at the start that one of my great loves is structural elucidation. It's a chemical equivalent of solving crossword puzzles, and it's something I like to think I'm still quite good at, and in fact better than most of the students. Sometimes I can still surprise them by coming up with a structure when they've been struggling. Polly.
Wait for a microphone here at the front, please.
So I'm intrigued as to why the organism would evolve, why evolution would give it this long and winding road.
Yes.
So there would have to be a quicker way in bacterial evolution to get to the end, but not cause catastrophe for itself. Is that?
Yeah, well, of course, all these reactions are enzyme catalyzed. And one has to remember that enzymes are just that. They are catalysts. They can't bring about chemistry, which would be otherwise unreasonable. And I didn't go into this in detail, but the formation of the ring necessarily involves an epoxide, which puts that extra hydroxyl group in. And we've now isolated the proteins which are responsible for that, because it in itself is interesting because it formed a 6-membered ring, where chemically it should form a 5-membered ring. And so there's 2 proteins involved there, one to control the cyclization and the other to control the ring size. So you're then forward with the issue of getting rid of the hydroxyl group. And there are many other examples of similar long and winding roads. So if you look at the biosynthesis of deoxysugars, where again you effectively have to remove a hydroxyl and replace it with a hydrogen. By and large, this is done by oxidation, dehydration, and then re-reduction. And that's essentially what's happening here. So overall, it seems a complicated, but chemically, it's an entirely reasonable one. So you oxidize one of the hydroxyl groups to a ketone. The hydroxyl group can then eliminate to form an alpha-beta unsaturated ketone, and then you have to do essentially 2 further reductions to get back to the proper ring. So it seems complicated, but it is actually chemically logical.
Thank you. Very nice talk. I wanted to ask you how you protect, for example, the discovery that you did for the synthesis or the biosynthesis of this last derivative, the C one. Is something that you can protect these genetic modifications, or is only the chemical structure, or there is a way to do it?
Well, the structures, in the case of pseudomonic acids, A, B, and C are known.
Unknown, yeah.
And these are, originally, they were Beecham compounds. They're now disappeared into the black hole, otherwise known as GSK. But, of course, the process we can patent. And so that's why I was being fairly coy about how we actually do it. And we're in discussions with GSK at the moment about how this can move forward. I have to say, I'm not interested in the money. I'm just interested, I mean, I like doing the science.
Yeah, but could become
Yes.
a very
Yes.
nice medicine. So.
Yes, yes. But yes, it is. And I seriously think that pseudomonic acid C will be, now that it's potentially available in quantity, should be a clinically useful antibiotic in its own right, which won't have the limitations that pseudomonic acid A has. So, you know, as well as getting the satisfaction out of the science, we can say, well, maybe we've done something to improve, you know, the lot of the public in general.
Thank you.
And Hannah, again, further back.
I was interested in the subtleties of the penta, hexa, and hepta
Yes.
ketide chains, and I was reminded of being surprised as an undergraduate of finding out the massive differences in hydrocarbon chains between the odd-numbered and the even-numbered. Is that the reason that they're done? Because it generates such a different physicochemical property to it, or physical property even?
Well, that's a kind of million-dollar question. Why do these organisms make these compounds in the first place?
Do you see those differences as you start to make each homologue?
Yeah, we can see differences in biological activity. So, for example, if you look at the mupirocin, we have analogues. It has this 9-carbon fatty acid chain there, and we've made both biochemically, biosynthetically, and also chemically analogues with chain length between 4 and 10. And the biological activity certainly maxes out around about 8 and 9. It goes longer, it gets less active, it gets shorter, less active. And if you can remember the picture of pseudomonic acid in the active site of the isoleucyl TRA synthase, it kind of curls back on itself. And that's suggestive that, you know, there's an optimal size to fit that cavity. But otherwise, you know, why does nature make these different compounds? They're of no obvious use. The Pennilyn systems do have various biological activity, and they inhibit certain ATP kinases, and there's some suggestion that it could actually be useful in treating neurological disorders. But, you know, that's a bit out of our actual sphere remit. We just like making the compounds.
Thanks very much.
And a little bit further behind.
Hi, I was interested in the A and the C.
Yeah.
Why is the C made at all? If A is made, it will decompose quickly and so not be toxic. But if you build up C, you're going to kill yourself, are you not?
No. The reason, if you talk about the organism or us.
Oh, I mean the organism, sorry.
Yes. I say what both A and C do is they inhibit the isoleucyl tRNA synthase of E. coli and Staphylococcus aureus. And what the organism has, it has its own isoleucyl tRNA synthase, which is resistant to it. So it has its own inbuilt resistance mechanism. And that's quite often the case with these antibiotics. The organism has a way of protecting itself from its own antibiotic. Sometimes it simply works just by pumping it out of the cell, or it has, you know, alternative proteins, you know, to the actual normal bacterial target.
So why has C survived? If it's not useful, why is it still there?
Oh, I think it's just a function of the pathway. I mean, we talk about enzymes being specific, but they're not that specific. So I think C just kind of leaks off the pathway. And what's interesting with a lot of these things is, you know, you look at a lot of organisms and the metabolites they're supposed to produce, and they're supposed to produce 1 or 2 major compounds. But with the kind of sensitivity we have now with extraction, and particularly NMR, we can see tiny amounts of compound. The analytical techniques are so much more sensitive. And without going into detail, if in pseudomonic acid, A and C, there is a whole string of intermediates. And when you actually start to look very closely, you can start to see these present, albeit in trace amounts, in the total extract. So they just kind of fall off. You know, they just, because the enzymes don't hang on to them 100%. Without going, these are all linked, or mainly linked to the protein through thioesters. and there are esterases present in the cell, so they just kind of leak off the basic synthase. Thanks.
More questions? Forward again. Sorry, Hannah, you're doing all the work.
I was wondering if you thought that the reason there hasn't been a successful MRSA antidote manufactured is because a single compound is isolated and extracted and processed without the numerous micro compounds with which they work synergistically in nature.
I think, of course, any drug has to pass certain standards of purity, and even when they're, again, in the sort of GPA process, there's inevitably impurities produced as a result of the synthetic process. It might be there in tiny amounts, but one of the conditions for getting drugs approved is you have to be able to reliably identify these trace amounts, but you also have to be able to reproduce them systematically. So if the process gives like 99.9% of the compound and then 0.1% of various impurities, you have to consistently produce the drug with these 0.1% of impurities there. Synergistic effects are always very difficult. I worked for a long time on mycotoxins, which are toxic compounds produced by fungi, of which aflatoxin is the main one. And it's quite interesting because when you read the reports on the isolation of mycotoxin, you'll start with a very toxic extract, but once you start to purify the compound, the toxicity falls off. And I think that's partly due to synergistic effects, but also it's just the matrix that they're in change, and so the absorption and delivery, if you like, to the active sites varies. So, I mean, that's the pharmacokinetics and all kinds of things involved there. I don't know if that answers your question.
Yes, thank you. I am also assuming that the reason that the pharmaceutical companies don't acknowledge the fact that it can be treated successfully with essential oils is because they make no money out of essential oils.
Can they be treated with essential oils?
They have been, yeah.
They have been, right.
Yes, several cases of MRSA
Yes,
have been
yes.
treated successfully with a
Yes,
mixture of essential
Yes.
oils.
Yes, well, I mean, it's like, take an analogy, a football analogy. Clubs like Rangers and Celtic are notorious for buying up and signing up all the promising young players from other teams and then sitting them on the bench so they can't play against them. And the pharmaceutical industry plays the same game. I mean, it's I believe you can design and build far more efficient combustion engines, but again, it's not in the interest of the motor trade to do that. So they have their own strategies and schemes for protecting their own products, of course.
Yeah.
And there is that element comes into it, of course.
Thank you.
Have we further questions or comments? No, I don't think so. Can I call on Professor David O'Hagan to propose a vote of thanks?
Thank you very much, President, and also Lady Black. And Tom, thank you for a wonderful lecture. Tom comes from the same tradition as myself. We're organic chemists, and as Tom's outlined in his lecture, this inspiration of natural products has always driven organic chemistry, and understanding the structure and stereochemistry and many of these natural products, these small molecules that come out of bacteria and fungi. We've seen today both bacteria and fungi. That distinction wasn't actually made, but it's very significant in its complexity. It's more difficult working with fungi, isn't it? So looking at this inspiration, looking at structure, has led Tom. I mean, Tom, all my career, has always been there. And what I mean by that, not just that I'm younger than him, but what I mean by that, what I mean by that is he's always been leading the field. He's always been taking the early sort of structure elucidation and total synthesis of natural products, which is an important aspect of organic chemistry and its sophistication. But as you want to really understand structure and get a deep insight into these natural products, which are really important because of medicines and all of that, then you also have to embrace new technologies. So new technology, somebody asked about techniques. So things have been advancing all the time. NMR, mass spectrometry, has really advanced. Molecular genetics has come into the arena relatively recently. And if you're trying to solve these problems, you want to embrace them. But that's not so easy to do. That's difficult to do. It's difficult to keep pace. And Tom's kept pace. He keeps it going. And he builds with them, with his enterprise and with his sort of focus, are people. And he is, you know, within our community, Tom is very well known for promoting people. And he's promoting people, the discipline, the interdisciplinarity, the broad base of people, not all the same type of people. People with different skills that can contribute to the problem. And he has been spectacular at assembling that sort of interdisciplinary team that can really operate at a world-class level. So he was slightly derogatory to biology at the very beginning. He said, it's always good to put one over on the biologist. I don't think he really meant that. I mean, you might, he's unifying chemistry and biology, and he's done that very well. So, Tom, thanks very much. I know you're going to retire from Bristol teaching, but you need to talk to Joe Conley, because this is just a transition. It's just a phase. You know, you're going nowhere. And we very much look forward to the next phase and the contribution. And what a wonderful lecture. Thank you very much.